Okay, good afternoon. So I'm recording this session for students that will miss this class. So last week, we spent some time to look for the right dataset for our project. So the next, I'm going to give you 5 minutes to reconcile, and then so that you have time to bring up MATLAB and also load your data in. Just make it ready, and we are going to start to analyze it. So let me know, and if any of you can share your screen and to show me the data you collected. We have to go through some quality control of the data. So after the filtering, see how many cells has left for our analysis. If you only have 50 cells left, not so meaningful to analyze them. So we need several hundreds or thousands of them. It will be perfect. If you download the 10x genomics data, you're supposed to have enough over thousands of cells for analysis after the filtering of the QC. All right, let me know where you are right now and how many cells you get. If you think you need to change the data set as an example, you may want to try again and send me the link of the paper so I can have a closer look, and I may help you to process it. I want every of you on the same page. You have your own data set to play with, have enough data, and high quality data. All right, 5 minutes for your preparation, and then we will get it started. Okay, anyone have any questions? Anyone want to share screen? I'm just testing whether my microphone has any problem. Can you hear me?
Yes, we can hear you.
Okay. Okay, so if you don't want to share your screen, I can share my example. And then because we have the recording and maybe later you'll just review what I'm doing here and follow the same procedure and see whether you can work out on your own data set. I'm going to use example data, which I can download from here. So in the chat, I point you to a GitHub link. This is a data set we generate our own. So this data set is from a cell line called lymphoblastoid cell line. To make this cell line, we just get a human B cell from the human blood. So you know the B cell is a primary cell producing antibody. So you can culture the B cell in the Petri dish. However, if you want to keep the B cell alive for many, many generations pass on, you can immobilize it. So you use EBV, the virus, to infect human B cell and transform it into lymphoblastoid cells. Those lymphoblastoid cells is a type of cancer cell, and it can propagate and pass on to next generation easily and easy to culture. So people use it to store genetic information. For example, if you want to sequence a population of cell, you may want to establish lymphoblastoid cell line for individuals. You collect the blood cell and keep it as a LCL cell lines. So you can do the genome sequence and the analysis on the genome sequence once you have a resource. Human genome is sequenced from these cell lines and assembled. Here in this study, we use lymphoblastoid cell lines as a model to study infection of virus to the host cell and to see how they interact in terms of their gene expression level in the same cell. Okay, because we use 10x genomics technology, we have information about gene expression of all the expressed gene in the cell. So this is lymphoblastoid cells. In the same time, we also sequenced this virus genome as well. Okay. So for many of these cells, we have virus gene expression at the same time. So that's the data I'm going to use. In this download folder, you download the zip file and zip it. There are two cell lines, one from a donor from a European ancestral background, and another one from an African donor. Both donors we know are female, and they are there's no information about their healthy status, so we don't know whether they have a particular disease or not. So we regard all this as a two human healthy reference. So their cells, B cells collected and infected with EBV virus. And then after the established of lymphoblastoid cell lines, we collect the cell and send onto this 10x genomics machine to prepare the cell and then get the single cell RNA-seq data. So I already processed the data and then get the expression matrix and also gene expression level. And here we have a MATLAB code, a piece of code to do this. But I'm going to just load this data set. So after I load it, I have this SCE variables. This variable is a compound variable. It's called cluster in MATLAB. It's called cluster. This variable hold other variables. So inside this SCE, we have .x, this is expression matrix. We have a G, which is a list of gene. That's essential information we need. We can scatter plot to see the sample, to see this data. That's on my screen. So if you identify the data set you want to study, and you collect the data, you download data, process in the same way I described in the documentation of the single cell RNA-seq analysis package, you should be able to get this three-dimensional embedding. So the first question you want to ask is certainly how many cells in there. There's basic statistics. We have 7,247 cells. And the number of genes we get information among the cells is 18,000, more than 18,000. That's a lot. So some of the genes will have a really low expression level, maybe only expressed in 1 or 2 cells, but we still keep there because they are sometimes informative. Okay, remember what we can do with this. I think you have previous lectures I discussed a lot using other example data. But here, once you have this, the first thing you may want to do is to, if you haven't embedded, if you have a cloud of cell with no clear clusters, you may want to click this button to embed the cell using either t-SNE or UMAP or FIT. But here I already computed this location of each cell. So I don't want to use t-SNE again because that will take some time. So this is a t-SNE plot basically. So I cancel this option. Okay, the first thing you want to do maybe is to classify them. So I'm going to click this button. So I want to cluster cells into different subgroups. So I select yes, and I use k-means. K-means is a quick way to do the first round of the classification clustering. This is based on the nearest neighbor, shared nearest neighbor. This will be more computationally intensive. So for this purpose of demonstration, I'm going to use k-means. And I have a rough estimation how many clusters I expect to see in the data. So I just use default in this case. I expect to have 10 subclusters in this group. And then once it's finished, the program asks whether you want to label these clusters. I can say yes, I use maybe index or text. Same. Now I have a name for each cluster. So look at the color is sometimes not good enough to differentiate them. Look at this color for group A, group 1, and group 8. They all dark blue, so I don't know. They're so close to each other. So that's why the label may be helpful. The 3 and the 10 are the same. And then you start to look at their this result of clustering. So basically, it's follow the similarity of groups. So you have an island of here, and then number 5, something here. Okay. If you are happy with this clustering results, then you don't have to do a justification. So, but in my example, I really think this small group here, can you see there are a couple of yellow or orange cells I believe should belong to cluster 8. So this automatically k-means clustering algorithm does not give me the best solution I expect to see. So I want to make some adjustment. What I can do is to brush. I want to select this brush and brush over these cells. So I brush some of these cells in class 3, some in class 8. Let's make sure you have at least one cell in that group, one cell in that group. The reason is I'm going to merge them. Not a good idea. I shouldn't merge them. Let's see. What should I do? So I want to find a way to reclassify those cells into group 8. Well, there's a way I can do. I just figured out. So first, I'm going to make a new cluster out of this selected group. So I'm going to select a small I'm going to select so this brush tool here, brush, select this group of cells. And then first I'm going to make a new cluster. So you see this button here, make a new cluster out of the brushed cell. And then I will update this ID. Now I have a new cluster, which is cluster number 11. And that's original number 8. So after this, so you see this cell I was trying to push into class 8 was in the cluster number 3, but now it's independently as a cluster 11. Now I can select cells and merge these two groups, which use this button here. So I'm going to merge brushed cell into one cluster. And I select yes, and I will select what is target cluster. Now I want to select number 8. So the group 11 will be merged into group 8. then update my class ID. So I'm done here. And I merged some of the cells from a group 3 was highlighted as in the tip of this into class 8 now. So that's some small adjustment you want to make. Obviously, there's a imperfect clustering, so you can make adjustment. So once we are happy with this cluster and algorithm results, we can do this export the saved data. The reason you want to keep your annotation, so you want to save your, export your data into this variable workspace. So I want to save this SCE single cell experiment object into variable named SCE1 because the algorithm detect there's existing variable named SCE, so it doesn't want to overwrite the old variable, but you can choose to overwrite it or you can keep SCE1. So in this case, I will save as SCE1. Okay, so you save SCE1 here, the same as what you have in this interface. So you keep your record. You can later save this as a file, so you don't lose any of the annotation you made here. Okay, the next step, you want to run a program to identify the cell type. You can do this for all the clusters. We have 10 of them here. I just click one simple button here. It will get the cell types for each of the cluster. It will have so I'm going to do it. Assign cell type identity to each of the clusters. Okay, yes. And this is a human data, so I select human and then use database, marker gene database, BanglaDB. Okay, the program will run and found the first cluster. So this annotation for the cluster number 1. Plasma cells. So if I say yes, so that's my annotation for cluster number 1. And then second, plasma cells. Third, plasma cells. Plasma cells are B cell, which actively producing antibody. So I know the cell type is lymphoblastoid cell line of my sample. So it makes sense for this algorithm to label all the cells in different clusters the same as the plasma cells. Okay, at the end, I have all the clusters has been labeled with their potential cell types. In this sample, it's a cell line, so it's a purified cell type. There's no differentiation of this cell type to any other cell types. So it's all the same types of cells. So I'm not so surprised to see this because I know the biology, I know the system. in your own study, you may have a different cluster of cells. And because you have your own original reference of the paper published for this data set. So you should be able to figure out whether the assignment of a different cell type makes sense, match with what you expect to see from the data. Oftentimes, they're inconsistent between their cell type label by the algorithm using the reference BanglaDB marker gene information. Because this is so complicated and some cell type may be not well annotated. So the information is not available in the marker gene database. You may not be able to predict cell type correctly. We see this all the time. So you have to make some adjustment, okay? So you for example, you have a cells from a lung and you don't want to, the first top annotation suggestion is from the liver cell, some particular types that are specific to other organs, not the lung, and you want to make adjustment. You may want to choose the second, the best hint of the annotation. Remember, I have a list of the candidate cell types you can choose from. Now I'm going to show you if you want to make adjustment about the name of the cell types, you should be able to do it. For instance, I want to rename this plasma cell in cluster number 2. There's a button, and I should be able to rename cell types, for example. You think you want to label them correctly, and you click this button. This button is next to the cell type annotation button. Then you'll see, okay, you want to rename a cell type, you say yes, and it asks you which group you want to rename. So I want to rename class plasma cells number 2. So I select this name and put in the real name, the B cell. It's still labeled as number 2 because it's number 2 clusters. So I press OK, and then want to relabel them as a text, the same. Now, the label of cluster cell number 2 has been changed from original annotation plasma cells, changed to B cell. So you can do this adjustment by yourself for any cell types you want to rename. Okay. So at the end, when you're happy with this annotation, you want to export the data, and again to save as a variable. Okay, so once you save this, it doesn't matter if you by accident close this interface, you still have all the annotation information has been stored here. You can restore it back by calling the scatter function and then put your annotated scE2 variable as your input. Here you get all the original annotation. And once you bring it up, you don't see the name, the label of the clusters, but you can label the clusters. Press this button. You can change which type of the cluster you want to use. But here I know it's from a cell type. So the cell type TX means it's text. and label with text. Yeah, so that's my my current data. I reload after I shut down my interface. I still have that annotation. You see the B cell annotation is still here. Okay, to visualize this, you can click this button. See, I want to make a video snapshot to make MP4 and this short video to help people visualize if other people don't have this MATLAB environment. You can share the MP4 file with them. Okay, at this moment, I want to know whether you have any questions and what you expect to do with your data, any suggestions we need to address here before I move on to another section. Do you find any bugs in the program when you run, you crash? If you do, please let me know, because I can fix this online, and you can have a new installation. You should be able to work out with a new version. By the way, if you don't like the color, there's a button which next to the refresh. So the last button here is a refresh. If you want to refresh everything and from the beginning, just as you reload the data, you have this option. And then the next button is to switch different color maps between different color maps. If you don't like this, you click and you change to different color maps until you found the right one for yourself. Okay, so next one, you can, after the color maps, you have a switch for different marker types. The reason I gave you these options, sometimes you have much more cells. You don't want to use this circle to visualize as a representative of each individual cell. You want to use just a single dot. Okay, so this option allow you to see more dense cells. If you have more than 10,000 cells, you may want to use this option to visualize it instead of each cell is representative as a circle. Okay, and also in many publications, or you may want to use 2D instead of three-dimensional. In that case, you can click this button to switch between three-dimensional representation to 2D. If I select this, by default view, that means you will keep the coordinates X, Y, Z as it is. And then to see the, by default, there's an x-axis, that y-axis. Okay. So by default is this is x and y, so you will get this. However, sometimes you don't want to default to the coordinates. For instance, if in this angle, you want to keep a projection of the cell in this particular angle, you need to select using the current view. And you will see, if I use current view, I will maintain the angle of what I'm seeing and project, scratch the data points instead of using its original x, y axis to present. I hope you feel the difference. For instance, if I use by default 2D visualization, I will not be able to separate this purple, gray, purple and the dark red cluster away from this dark blue, because they superimpose onto each other in our by default 2D coordinates. However, if I want to view the difference between these two groups, I want this particular angle to be kept. So I will select current view projection, and that's my new 2D presentation of the cells. And you can see the separation of these three clusters. Okay. As I said, you can embedding your data using different approach. And by default, we are using t-SNE. And if you decide to use UMAP or FIT, you can certainly click on this button. I'm going to click a FIT and see what's the difference, because I still have the color labeling. I want to see whether the same color clustering based on the t-SNE coordinates will still be clustered at the same group in the FIT embedding. You see, I click the button, and in the background, the FIT algorithm start to run. And there are iteration procedures in the algorithm. So you see the progress until it reaches some level of the converge, it will stop and present the data. So just give it a couple of seconds. This algorithm is fast enough to give results very quick. All right. So FIT gave this slightly different view of your data. And remember, they are the same set of data, but you just use different ways to present, and you get different results. Looks like FIT gave a huge cluster in the middle, and then there's 3 branches, 2 or 3 branches. Okay. All right, so if this is the real relationship between cells, then for this particular shape, I will be able to do something different. Okay, the t-SNE give you the clusters, big one, small one, but here give you some of the trajectory-like shape. So I'm going to run a trajectory analysis with this data set. So let's first, the color doesn't mean anything for me right now. So I'm going to get rid of the color, change current class tab. So yes, if I use cell type, oh, I still have it. I want to get rid of with batch ID. Yeah, I have no way to get rid of this the color. You know what I can do is to I certainly can merge merge all the cell types into one cluster. And then merge that again. Okay. So I use a merge because it doesn't make any sense for my data set to make subgroups because they all LCL, they're all the same cell type. Okay, so I just merge everything into one cluster. The reason I clean up this view because I'm going to run a program which is this. So I'm going to build a trajectory. So this algorithm, if I click, for instance, I'm using splice feed. This algorithm search the structure of this representation and found the longest path from one end of the structure to another end. So in this case, I start from the very beginning as a starting point on your right, on your left, and search against the cell and make a trajectory in the right curve and end up with at this end. So making a very simple trajectory analysis, which is just based on the location of cells, nothing else. And if I assume this population of cells start from here and then developed or derived or maybe change the states along this trajectory, and to the right end, you have more cells in much different states as your starting point, the cells here. Okay. So this trajectory is built purely based on the neighborhood of those cells and try to find a path and to assign the cells along this path to the main or the spline fitting line, okay, in this line, and you can follow the trajectory from the starting to the end and give each step a time point and assign cells from starting to the end, different time points. Okay, this is called pseudo time. It's not a real time point because we didn't sample the cells at a different culture time, but we just reorder the cell using this nonlinear, the curve, to represent the potential time point at a different stage of differentiation and assign cells to its nearest time points on this curve. So every cell will be given a pseudo time point. I don't know if I explained this clearly enough. But for this analysis, we use this red curve as a major trajectory. So we ignore this branch. So this cell will be still assigned into this trajectory and give a pseudo time. Okay, I'm going to take a 5 minutes break. And we will continue to see how can we improve this and what that means here. Okay, the expression level change along the pseudo time. So you can collect genes and see which gene change most, follow the trajectory of the time. Okay, let's come back at 3 o'clock. Maybe 3:05. Okay, I take a 10 minutes break. Any questions before we leave? So for this trajectory analysis, once you build the trajectory, every single cell has been assigned with their pseudo time. And then you can follow particular expression level of the gene along the cells, along the trajectory. And then you build a profile of gene expression level averaged across the cell in the same time points, and then look at the change of this gene expression level of certain genes along the pseudo time trajectory. So what you see here is a set of genes show potential interesting because their expression level as a function of the pseudo time, and their change can be detected. So we start from time 0, pseudo time, which is the time point on this plot here. And all the cells in this group, if we look at the green gene, which IGLC2, all the cells in this group have a relatively high expression level to the level of normalized like 12. And this pattern, the ink get decrease. This expression level mean decreases with the increment of the time, pseudo time. So what that means is if you found a group of cells in the middle of this trajectory, then its level of expression of IGLC2 will be relatively low, lower than the starting point. When you reach to the end of this trajectory, then the average expression of IGLC2 is only about the half of this at the beginning of the projection trajectory. Okay, you see the decrease. Similarly, you found a gene called IGHA. This decrease as an increase of the pseudo time. And the light blue gene, GCHAIN, decrease as well. In the operate chain, these 4 example genes get upregulated across the time. This include the first orange one is TMSB4X, and then this blue is ACTB. And then there's other 2 genes. So that's the purpose of the trajectory analysis, try to identify the gene which may associate or drive the cell differentiation stage, because they show increase or decrease expression level across cells as a function of pseudo time increment. Okay. So what I can do is next to highlight some of the genes expression in this plot. For example, if I choose to look at IGLC2 expression in this cell, which I can do this by selecting the first icon here. And I want to show the expression of single genes. So the gene names IGLC2, which is this gene. I click OK. And you see this. There's 2 views of a single gene expression. So you see expression level in this tip is really high, because if you look at this color bar, it indicates the gene have a really, really high expression in this tip, and it gets decreased gradually when you move to the end of this cloud. Okay. Similarly here, you see the expression levels given by this z-axis. So you see high expression level of this gene in the tip here, and they change gradually into lower, lower, lower. Okay, so I can roughly conclude the fate gave you this long branch, that because some of these type of genes driving the cell looks so different from other cells. So this IGLC is immunoglobulin, a constant chain maybe. Okay, we can try another, which is PFN1. Okay, single gene PFN1. Get this plot. Okay, you see it's clear they show high expression level in the middle, which is right here, which that's end of the trajectory in our definition. And if you think that's a starting point, then you have an increased level of expression, follow the trajectory. So that's PFN1. Okay, we can combine the signal. You can, if you select, see, I want multiple genes to be combined. In this case, I will select multiple genes. I will do IGL C2 IGHA1 and the green one, the blue one, GCHIN. Yeah, and so I will combine the expression of the 3 genes together. And once it's done, you see this combined expression gene, 3 gene plus combined the pattern. Okay, so it's clear to me this trajectory, this long arm created by the feet was driven by some of these key genes, make them least similar to cells in this bottom, the base of here, the group. Okay, so how if you have 2 arms? There's one way you can make this results better is to remove some of the cells. So next I'm going to select these cells along this arm and going to remove them, exclude them from the analysis. So I highlight those cells. Okay, so I select, I brush them. I can delete the cells by click this button. Delete selected cells. Select cell will be deleted. Let's confirm again. Okay. So now we have a much clearer trajectory cells along the trajectory. I may want to exclude some of the cells in the middle because so I just exclude them temporarily from the analysis. So that's my clear cell map, and I want to do the trajectory analysis again. You know, you have 2 selections. You can choose either spleen fit or this called principal curve. So I'm going to try principal curve this time and see what kind of trajectory I can get. Ask me whether to show the expression of select genes. Okay, see. Yes. Well, this time the pattern is more complicated because this algorithm will follow so vigorously along this trajectory and introduced a very complex trajectory. But maybe this is overcomplicated. And then the cells, the genes select as example are those blue one, still this, the green one, see there are some other genes have been selected. So from seeing this trajectory, I think this analysis is over complicated, and we probably don't need such a close model of trajectory. So I will change back to spleen fit. really should clean up the data first. Yeah, I should clean up the data first, then select this split feed. And also, the absolutely starting point is not so important. So you can use this end as starting point. You flip the time. If you choose, if you know this should be the starting, and that will be end. Okay, so same results, basically. We got one more at the DKG and bump up. If you have your R package installed, you can use monocle, which is another method to build the trajectory and the pseudo time. I have that installed, so I can click and run. But if you want to know how to install that, read the package of monocle, which is an independent third-party software package in R. So if you click this button, this function will call that function. This application will call that function to generate the pseudo time. But I'm not going to try this because that will be slow. Now, I'm going to re-embedding my data back into t-SNE. Do you have any questions, any of you? We all good? Good. So next, once we finish this embedding, I'm going to show you in this particular data set, you can look at the various transcripts expression level. Because we process the data, not only look at the host gene, the human cells expression level, but also the various expression. So I don't know how long it will take, so I will give up and just load another set of the expression. Oh, it's almost finished. Okay, so it's finished. So, okay, in this session, I'm going to show you the expression level of the various. So look, so that's the host cell, and then if you found the gene expression in this list, at the very bottom, we have information of the EBV various expression. I don't know which one I should look at, but I select this one. You see this gene from the virus, it's called EBV BALF1. Okay, F1 expressed in 4.47% of cells expressed. You can detect its expression in 295 cells. Some of cells have this level of expression. We can try to combine some of these EBV virus because the expression level may not high enough. So if I combine this couple of EBV virus expression, you see 4%. This bunch of EBV virus, so you see 33%. So if you try all the EBV virus transcripts, almost every cell have at least one EBV virus transcripts expressed. Okay. Now, my next question is, can we build some kind of gene regulatory network using the genes? So if you decide to do this, you can click build a gene regulatory network. So you click this and by select the genes. So if I, for instance, I'm interested in EBV virus expression transcripts. So I can select a couple of genes from EBV. And this will create a network among EBV transcripts. It's not a very good view. Let me change the layout. Don't think this is correct. Let me select another set of genes. So let's try human genes. There's something wrong with this, so I have to figure it out. If you choose the list of genes like this, paste your genes in there, you should be able to get a gene yeah, I think there's a problem here. Let's try this one. Yeah, I select the gene, but it does not show up in my network. So I have to check my code. So don't work on this yet. There's something wrong with here. Okay. So I'm going to go back and fix this bug, and for the gene regulatory network construction, I don't know why there's a problem here. It's supposed to construct a list of genes. You select a set of genes, the network will be for that set of genes. But it looks like the program right now just gave you a 100 gene as an example. So something wrong with in the middle. So I will double check what's going on. Okay, for the rest of the 30 minutes, I will leave it for you, and let me give a quick summary. So I use one example data to show you how to do the clustering after you have this cell embedded. And how are you going to annotate each cluster and give a potential cell type annotation. But before that, I show you how to make adjustment for your automatically clustering algorithm result. You want to reassign some of these cells from one group to another, or you want to make a new cluster, or you want to merge some of the clusters into one. So once you are ready, and you can annotate them. And again, it's not a guarantee the database will give you the right answer. So you have to look at the context of your paper, the system these cells have been generated from. And then I showed you how to save the annotation into a new variable name. So you can save this into the file later. Don't lose it because you put your efforts in annotation and you don't want to shut down, close the window, and then the annotation information gone. It's not what you want. You want to keep this to save them for later use. Now I showed you instead of embedding cells using t-SNE, you can use other options like fit. And the fit tend to give you a global structure contains a lot of arms in the cells population that allow you to use pseudo-time trajectory analysis. This to assign a time to each cells based on their distance to the trajectory you just built. With that information, you can identify some genes with expression associated with the change of the pseudo time. And at the end, I've tried to show you how to build a network of genes, but I found a problem in the program, so I'm going to fix it. And I will let you know, and you should be able to download a new version of this and should be able to do it. Okay. So I leave you for the question, and I'm going to fix this bug on fly right now. I can see what's going on. Okay. Let me know if you have questions, and when you play with your own data, you may have different problems. And I hope you can try out those options and figure it out yourself. Or shoot me an email before this week and next week. I don't know, next week you should have a good progress and have something to show and have a coherent story why you choose that data, why the study make you feel interested in, and you download the data where you get it, and how many data you get after the quality control, what the shape of your data population, cell population. Have you tried any of this analysis? Anything consistent or inconsistent with the original paper? So you should have a rough draft, this picture, some of the slides maybe already there. So I'm trying to see what's going wrong and with my network building function and why it's not calling the right list of genes. Okay, got this. So good. Okay, if you don't have any questions, feel free to leave, and I will let you know once I fix the problem that you can re-download the package. You should be able to run the program to construct a network.